Bismillahirrahmanirrahim. Assalamu alaikum. Hope all of you are fine. I welcome you to PMDC online lecture on polymerase chain reaction. This topic is from biotechnology chapter of second year. We will learn the basics of polymerase chain reaction, its mechanism and its uses. Polymerase chain reaction is abbreviated as PCR. It was invented by Carey Mullis in 1983 and he got Nobel Prize after 10 years in 1993. Polymerase chain reaction is also called molecular photocopying. It is a method of DNA amplification or simply a method to clone the DNA template for generating multiple copies. Have a look on basic components needed for PCR. Together they form PCR mixture which is placed in a thermal cycler. First, DNA template, which is also called target DNA. It is the desired segment which is needed to be cloned. Secondly, two primers are required, forward and backward. They are required for working of DNA polymerase. DNA polymerase, it is an enzyme which is also called Taq polymerase. It is thermostable, that is, it cannot be deactivated by high temperature. It is obtained from bacteria called Thermus aquaticus. Then 4 nucleotides are required: adenine, thymine, cytosine, guanine. Buffer which contain magnesium. The process of PCR is divided into 3 stages. First one is denaturation, during which unwinding of target DNA occur. Annealing, it is joining of primer at 3' end of DNA. And last one, elongation, that is polymerization. It is the addition of nucleotides with the help of Taq polymerase resulting in complete DNA. I represented whole process in this table, making it more clear and easy to learn. You can see 3 steps: denaturation, which require 94 degrees Celsius temperature, then primer annealing, which occur at 54 degrees Celsius, and extension required 72 degrees Celsius. During denaturation, double-stranded DNA are separated into single-stranded DNA by the breakdown of hydrogen bond. Then primer annealing, attachment of the primer to dash ends of DNA strand. Can you make a guess at which end primer are attached? 3' or 5'? Okay, the correct option is 3'. In last stage, extension, new nucleotides are added at 3' end of the primer, completing the DNA strand. PCR depend upon series of 20 to 40 repeated cycles of DNA replication. After each cycle, the number of DNA strand is double, and at the end of whole process, 1 trillion copies of DNA are obtained. Diagrammatically, you can see here, dark blue color, it represents the template or target DNA. During first stage, that is denaturation, the template, they are separated, double-stranded DNA becomes single-stranded. In second stage, annealing, green color primer is added, and note they are added at 3' end. And during extension, nucleotides are added and complete DNA molecules are formed. Few applications of PCR are listed here. It is used to study the DNA, and detection of pathogen and diseases is possible by PCR. And last but not least, it is used in criminology. Now the summary of polymerase chain reaction. You can see the thermal cycler which consists of PCR mixture containing Taq polymerase, DNA samples, primer, etc. Here one cycle is shown consisting of denaturation, annealing and extension stages. Here is an assignment for you. First question: if the PCR is in vitro DNA replication, in vitro means which occur outside the cell, then which process is called in vivo DNA replication? Second question: how many cycles of thermal cycler is required in PCR reaction? Wish you all the best. Thank you so very much.